Okay, so today is Bring Your Own Data Day, and the exciting thing about that is you guys have brought, I think there's like 16 or 17 of you here right now. You've probably got like 22 different types of data, so we're excited to help you through all your data. So the way today is going to be organized is we will have a methods lecture on data processing, and then if you guys need help with data processing, I think we have 2 hours reserved to help you with that. Of course, it might bleed into the next section. The next section is on model engineering. Oh, let me take this off. The next section is on model engineering. And on the model engineering section, what we're going to do is walk you through how we might approach taking data and building, an intro neural network, or, how we would go about something a little more complicated than PCA. And we have wonderful TAs around the room. When we start the section, I'm going to have them stand up and introduce themselves. So if you work on single cell sequencing data, you are very lucky. You have many single cell sequencing data people here today. You also have Matt, who works on not single cell sequencing data. So we will all walk around and help you, and feel free to ask us questions. Okay, so let's start with the data processing methods lecture of the day. And so we're going to start with data processing principles. And this is by no means exhaustive, but we're going to walk through 5 that are very important and that you should make sure that you're checking, no matter whether you're dealing with sequencing data, imaging data, time series data. It pretty much goes through all of neuroscience data as well as most data just in general. Okay, so the first thing I want to ask, and this is a question for you all, and this is going to be a very interactive lecture, is what are your data processing principles? I know you guys all work with your own data. You maybe don't have them codified in like a list of every time I get data, I'm going to do X, Y, Z, but I'm sure you guys have some intuition for like what you do and how you go about it. So what are your principles? Visualize, okay, that's a good one.
Pre-registering what counts as kind of usable and not usable data.
Okay.
And part of that is, if they're an outlier to the distribution.
Okay, so data cleaning. So pre-registering, data cleaning, that's a good one. We're going to get to at least 3, so someone else has to speak up. We've got 5, so I know there are at least, 15, so I know you guys still have ideas. I'm not going to take it from the TAs, but if it becomes drastic, I will call on you, Sina. No one else? All right. Waylon, what's one of your data processing principles?
So one part, I guess, belongs to data is denoising.
Okay, denoising.
Yeah, some physiological artifact that we want to look at.
Okay, that's good. So far we've got visualization and denoising and different types of denoising. So we're kind of diving into how denoising means different things for different types of data. How about JU?
Try to preserve your original data as much as you can when you clean it up. Although I haven't personally been doing that because I wanted just to get to 80 or 90% of that working and see what results we have.
Okay.
Fixing all of them takes a very long time.
Right. So JU also brings up a good point, which is this idea between, I know that there's more data cleaning to be done, but there's also this idea of getting to a good enough model that works. And so something that we're going to see again and again today is there's going to be some data processing, and then you're going to model it, and you're going to see I didn't capture something, and you're going to need to go back and fix something about your data processing pipeline. So all of these are good ideas, and keep thinking as we walk through this list of 5, there are certainly more, so think about what we're missing and what we could add on as well. Okay, so moving on, these are the 5 we're going to cover today. The first is visualize your data, and this really helps with intuition development. So when you see your data, try to look at it in different ways. Try to understand what your data is telling you and visualize it as many ways as you can. The next is signal extraction, which is also known as denoising. This is another important piece, and there's different types of denoising that you can do. And I'm sure for all the different data sets, like from flies all the way up to humans, you have different tools that one might use to do this denoising. Another is data set augmentation, which is something you guys might not be as familiar with, but is very important, especially for those of you that are going to deal with imaging pipelines when you have images of data sets or data examples that are not very well represented in your data set, but you still need to be able to do work with that. All right, the next is normalization or standardization. And again, this means different things, and there are different standard practices for different types of data sets. And the last is train validation test splits. And so earlier today, I was actually chatting with Matt, and he was like, oh, you're going to present that in data processing? And I'm like, yeah, I think it's more data processing. And he's like, no, I think it's more model engineering. And so the point that I want to illustrate with this is that two people that work with this a lot, we have different perspectives on what counts as data processing and what counts as model engineering. So even though I'm presenting these concepts in two different lectures, recognize that they are very fluid and they're going to need to communicate with one another. So think about how do I need my data to look to be able to model successfully, and vice versa. So here, you'll notice that none of these specify a specific type of data, because I personally work with time series data. Sina, Delaney, Christian, they all work with sequencing data. Matt might work with imaging data. He might work with synthetic data. There's all different types of data. And so what we're trying to do here is pull out principles that are going to relate to all the different types of data that you guys work with, which are going to range in that spectrum, all the way from time series to images to sequencing to behavior, so many different types of data. So this is not exhaustive, but we're going to try to pull out principles. We'll walk through each one of them with an example in one of those classes, but definitely think about, if I present something for time series, how does it relate to images? If I present something for images, how does that relate to single cell sequencing? So we're going to try to do the most exhaustive job we can, but we're definitely not covering all the principles, and we're not covering how it relates to all types of data we might encounter. Okay, so let's dive into visualize your data. And so I've put this image on the screen, and I have not told you what this is, and I realize that you're going on lots of partial information, but given that this is a neuroscience summer school, I'm going to ask someone what they think this is. And wrong answers are good because I've told you nothing, so I don't expect you to have the right answer. Shout out guesses, literally anything.
Maybe cells in the brain.
Sorry, cells in the brain, you're saying?
Yeah.
Okay. Yeah, so that's definitely...
You could draw something around it that looks like a brain?
Yeah, so it looks brainy, all the things together, they definitely look brainy. And so Gregorios is right, this is a brain. And you have lots of different colors, right? But there's some of the same. You have lots of orange dots, you have lots of green dots, you've got some purple stars, some aqua stars. So I'm just going to cut to the chase and tell you guys, since I know it's early, but you'll get more talkative, I hope, as the day goes on. This is a brain. This is a human brain. This is a human atlas brain. And what we've done is we've taken electrode locations from this human brain, and we've put them all into the same representation. It's actually the MNI Brain Atlas, so Montreal Neurological Institute Brain Atlas. And so what you're looking at is 12 human patients whose electrode locations are all in very different spots, and they don't really share too much. But when you put them onto the same atlas and you visualize them together, you get the representation of what a brain looks like, left hemisphere, right hemisphere, even though most patients only have it on one or the other. Now, as you might imagine, electrode data in humans, it's hard to come by, but also you want to get as much data as you can from it because you have them implanted for oftentimes a very short amount of time. So for each of these electrodes, you're going to have multiple days worth of recording. For each of these days, you're going to have an order of 100 electrodes for each day. And for each of those electrodes, you're going to have multiple time series instances for each of those electrodes. So you're looking at a huge, huge, huge amount of data. And so this is probably one of the most simple visualizations you can do, which is just spatially figuring out where does everything lie on top of one another. And I would encourage you to always go for the easiest visualization first with your data. Of course you can look at all your time series, of course you can look at representation of your cells, but just go for what does it look like spatially? And that helps you kind of register it in your own brain. And so I mentioned this already, but just to put it on the screen, and this is actually a picture of my whiteboard from a few months ago when we were trying to think about like how should we think about this data, how should we organize it. And so in the first column, you have different patients. So in this case, we have 12, patient 1 all the way through 12. Now for each patient, you may have many different days of recording. So you'll have like day 1 all the way up to day D, and sometimes that goes up to like 7 days worth of recording. For each day, you're going to have an order of 100 electrodes for each patient. So you're going to go all the way from electrode 1 through like 50, all the way to electrode E. And then for those, each individual electrode, you're going to have multiple time series instances for like every single electrode. So you're looking at a huge amount of data. So what I want to ask you is if I was to do it, and if you guys were to do it, how would you want to visualize this data first? So let's say maybe say second. I've already visualized it for you spatially. What is the next step you would take? So you know you've got patients, each patient has days, each day has electrodes, each electrode has time series. What would be your first step? Okay, so you want to look at the time series first. Okay. And so that's a good intuition. You want to look at your lowest level data first, what you have. But how are, yeah, Mihai.
Sorry, I don't think I understand. When you say each electrode has time series, do you mean like per stimulus or per condition? Or per
Yeah, so this data set, that's a good question. This data set is naturalistic. So there's a few ways that we can actually break it up. And just for those of you that do not have your own data today, this is the data set you are going to be working with. So if you don't bring your data, definitely pay attention because you will be working with this shortly. So there's a few different ways that we can go about thinking of the time series. So here, all of these like data have been collected in naturalistic settings. So there isn't necessarily like a action that's being asked to perform or like a behavior or a experiment on the participant. So one way people have processed this data is gone in and taken about a minute and a half and given it a label. So like, was the person sleeping? Were they watching TV? Were they eating? So I would count that as a time series. Another way to process this data, which is the data that you can work with today if you choose to, is people have looked at the arm movement. So like, is the arm moving or resting? Because a lot of these patients have like their electrodes over motor cortex. And so think of a time series as either in a naturalistic case, like a minute and a half of behavior that is given a label like eating, watching TV, etc. There's also a more granular like label you can give where like, is the arm moving or resting? So you can think about it in either of those two ways. Okay, so let's go back to Nayon's point about let's visualize the time series first. And that is a good point, good intuition, and that is where I would go first. And the reason isn't necessarily because it's going to be useful once we process the whole data set, because as we see, visualizing a time series, visualizing 1 or 5 or 20, that's not super helpful because you literally have thousands of time series. But you want to give yourself some intuition for like, what are you looking at and does that make sense? So try to go low level first, right after you've done like the most high level thing you can. Okay, so good idea, go to time series. How are you going to go to time series? I could visualize a time series for every patient across the first day. I could visualize a time series for electrode 1 for every patient, across all days. Like, how are you going to visualize this time series? Because there's many different, let's say, indices of this array that you could index, or this tensor that you could index to visualize this time series data. So how would you guys do it? Yeah.
The objective here is to reach that visual.
No.
Or what's the goal?
So the goal right now is for you guys to visualize your data in a way that is going to give you some insight into what you're looking at. So this visualization on the left, which is all of the electrodes mapped onto the same atlas in the brain, this is one of the most high-level visualizations you can do. And since you've gotten this high-level visualization, the next thing that one might think about is how can I get a low-level visualization? Because I've told you that you have time series on electrodes. You guys have no idea what these time series looks like. So it makes sense to now go look at the time series. But there are many different ways to look at the time series. Do you want to look at all of the time series for a single electrode across 5 days? Do you want to look at one electrode across all patients for the first day for every patient? Like there's many different ways. Let me actually do this on the cursor so Sam can see too. So for instance, I can look at one patient and I can look at one day, one electrode, and then I can look at all the time series. Or I could pick all the patients. I could say, I only want to look at what day one looks like in all patients. Like maybe there's habituation in this behavior and I want to see what it looks like on day one. And then I look at all the electrodes and all the time series. Like there's many different ways that you could operate throughout this stack to get a time series visualization.
Well, I mean, wouldn't it
Yeah.
depend on what your expectations are for, what leads to the greatest amount of variance?
Yeah, so I haven't said anything about modeling yet. I haven't said anything about trying to understand variance. All I'm saying and all I'm asking is how would you visualize this time series? Like I have just handed you this data set, which for some of you is the case today. I literally am handing you this data set. What would you do? Because today you are not given any code. You're gonna have to do this on your own.
I mean, I would do it across patients by day.
You would do it across patients by day, okay? Does anyone else have a different answer? Yeah, Mihai.
Across patients by day initially, and then depending on the task at hand, maybe look at clusters of electrodes.
Okay.
For example, if patients or subjects were looking at the TV, maybe I'll look at the visual cortex. If they were doing something cognitively demanding, I'll look at the prefrontal cortex, I'll group electrodes based on the task, and look at it on different days. Or if I don't care about time, I would just aggregate across days and then separate subjects according to other demographic or whatever behavioral characteristics those subjects have.
Okay. All right, so this is a good intuition from both Nina and Mihai that they want to see things across patients and they want to see things across days. Very good intuition. But I did say that this is like 12 patients across sometimes 7 days for each of these 12 patients across, so like when I say a day, I mean literally 24 hours of recording, across 100 electrodes for thousands of time series. So I would love that too. This is a terabyte of data. You can't do that. So what would you guys do? Okay. In the interest of time, we're going to keep going, but this is a good discussion, and you should always think about how can one do it. And then, yeah, Nina.
It doesn't just depend on your question, right? Like I assume you wouldn't just collect all this data or these data without, having maybe an interest in a particular function or a particular region.
Sure.
Wouldn't that reduce the space in some sense? But I guess what you're saying is that would be kind of a misuse of the data because you actually have all this sort of data. So why would you?
So I think you're bringing up an interesting and important distinction for all of us, which is the idea of data-driven like engineering versus model-driven and hypothesis, or data versus like hypothesis-driven. So like, do you want the data to define your hypothesis, or do you as a scientist say like, oh, I've got these electrodes in human, they're all over motor cortex, I think that they're gonna have this, and I'm gonna go look for that. So right now we're gonna go more with the approach of there is no hypothesis because you guys don't know too much about the data. We're gonna try to see like, what can we see in the data and what can we like extract from it? Okay, but all good points. So what I would do, and given the size of the data, given the scope of it, and given how much there is, one of the first things that I would do is I would say, let's forget about all the patients. Let's just pick our best patient, and let's see what our best patient looks like. And so I'm gonna pick the middle patient. I don't know if that was me. And then I'm just gonna pick a day. Like a day already has order 100 electrodes. That is a lot of data to visualize by itself. And so then what I'm gonna do is I'm gonna pull out, or rather I will pull out, but just so that you guys know, this green electrode map that I've plotted is the same as the green one in like the full patient representation. So you can see how like this is the 12 patients all on top of one another. If I just want to plot one of the patients, that's what their electrodes look like. So once I've picked a patient, once I've picked a day for that patient, I'm gonna go ahead and pick an electrode for that patient. Because even for a single patient on a single day for a single electrode, I can still have thousands of time series instances, which is not an insignificant amount of data to process. So once I've done that, what I'm gonna go ahead and do is I'm gonna visualize all the time series. And so we're gonna keep visualizing our data. And I taught you guys the trick of like, okay, you can treat your time series data as like as an image. And so here what we're plotting on the y-axis is the instances. So in this behavior, you've got, let's say, move. You've got roughly 400, I think it's like 415 instances of like move. So I'm plotting each of those instances across time. Who's happy with this visualization? Okay, why are you shaking your heads?
What's the bar?
Yeah, so there is a color bar, which is good. So, but yes, it would be good if I had put that. So yes, what is the intensity? What does that actually mean?
I think the main problem is that it looks like nothing.
It looks like nothing. Yeah. So you can't really tell much from this. And part of that is because like the ranges are very large. Part of that is because like we're trying to plot a lot of data and we're trying to visualize it. And because we're trying to visualize so much of it, you can't tell anything about any one instance of it. Okay, so this is soup. Another thing that I can try to do is I can try to plot a histogram of the actual values inside the array, and I can see if that gives us anything. And so that's the histogram. So the counts of like the actual like intensity values inside this array versus just how many like of these values are. Who's happy with this histogram? Okay, yeah.
That's okay, it's a fine histogram, but I guess the main thing is like, do you lose the time aspect? It's I guess,
Yeah.
where intensity is.
So you definitely do lose the time aspect. The thing I was going for here, which, and I like the way you put it, which was that the top image was soup. By plotting a histogram, sometimes it's useful. You guys have already seen instances where it is useful. Here, it doesn't get me anything. It's still soup. Like nothing has been elucidated by plotting it in this way. So like the top visualization wasn't particularly helpful and the bottom representation wasn't particularly helpful. So we have to try something else. And so there's still, even though like we've whittled it down from like the 12 patients that we had, the 7 days that we had, and like the 100 of electrodes that we had, we're like literally plotting one electrode and all of its time series instances. This is still too much data. And so one of the things that we're gonna, actually I'm not gonna tell you, what is one of the things we should try next? This is too much data, what are we gonna do? Okay, so we can reduce the dimensionality, and we're going to get to that a little bit in the model engineering lecture. Zoom in. Yeah, let's look at less of it. All right, so now what I'm doing is I'm only taking 25 of these instances, and I'm plotting it against time. What do you see, and are you happy with it?
Still doesn't say anything.
Okay, so Mihai says you don't see anything. And I agree, I largely agree, this is largely soup. If I was to look at this though, one thing that I'm gonna look at is this line right here. Everything else seems to kind of be doing something, but this seems to not really be doing anything. But it's not very satisfying, and you're right, it's not super satisfying. So since we've determined that this is soup, I'm gonna go ahead, I'm gonna plot a histogram, I'm just gonna see what happens. And indeed, as we've already established, it just gives us nothing. Okay, so I've tried to visualize this data. It's too much to visualize at the start. I try to zoom in, and it's still not giving me something helpful. As we've alluded to, this is time series data. We have multiple instances of time series data. What are you gonna do next?
What's the histogram?
So the histogram is, so let me, where's my cursor? Okay, so each of these, so this is like an instance of neural activity, like electrode, LFP activity, local field potential across time. The histogram is each of these like LFP activations has some intensity to it. So like how much voltage, bulk voltage activity was there. And that's what the color bar corresponds to. So this histogram is plotting all of the values of this array as a histogram.
On what bin? What's the bin?
I don't remember what I did. Yeah, the bin isn't too important in this case. It is sometimes. In this case, what I wanted to see by plotting this is like, do I get something interesting? Like, do I get a multimodal distribution? Do I get like 2 peaks that shows me that like there's 2 different activations? But by plotting the, I kind of gave you the answer. But by plotting the bins, I'm just seeing that it's basically like a Gaussian, and that doesn't tell me anything interesting. So for this, you're right, having the bins is important. For this, it's not so much like this particulars, it's the fact that the distribution is not something that helps us tell us anything about the data.
I mean, yeah,
but if you bin it maybe like smaller bins, maybe you get distribution that's
Yeah, that's true. In this case, it does not do that, but yes, that is true. Okay, I kind of gave you the answer already, so we're just gonna go to it. What you can do, which is a very helpful technique for time series plots, is called a waterfall plot. And so here what I'm doing is I'm taking the 25 instances that I zoomed into, and I'm just plotting them. So they're plotted on the same time axis. So this bottom piece right here, it's the same amount of time. And this one, it's still the same amount of time. All I've done is I've shifted it over a little bit to make it able so that I can see it more. And I'm just plotting them all. So all of these 25 are these 25. And what you see is that this thing, this neuron right here, not neuron, this electrode right here that gave me something that kind of looked questionable, but I couldn't really tell, is now very clearly seen as something that's a problem. And so what I want to illustrate here is that there's one, you're going to have a lot of data. The only way that you're going to get any intuition about this type of data is by zooming in on it and making sure that you know what you're looking at. And moreover, once you zoom in, not all visualizations are going to be equally helpful for figuring out what you're looking at. So definitely try to make sure to zoom into your data, look at the smallest piece that you can that's reasonable, and once you have some pipeline for processing this, make sure to include it, because you cannot look at a terabyte worth of data with your own eyes. You can't do it. And so if I was looking at this and I'm doing my data exploration, I'm going to see, okay, there are some instances in this data set where I just have like a line of a value. And so what I might do when making like a data set processing pipeline is like, if I have 50 instances, in this case, I believe it's 0, but we can go in and check, and some of you will go in and check. If I have 50 instances or 30 instances or 20 instances of everything that is 0, I throw away that data set example. And what I want to illustrate is that you're not going to be able to look at the entire terabyte of data. You just can't do it. So look at pieces of the data for every patient on like every day, if you can for every electrode, but even that is asking a lot. Try to pull out principles of the data that are problems or work. And then you can code those in, and then you won't have to look at all the data.
Do you know it's a technical and ontological?
How do I know that it's, what was the first word?
A technical problem.
Yeah. Okay, so that's a good question. So part of it is because it's like human neuroscience data. And oftentimes when you have, so all of the patients that are implanted, they're epileptics. Like you can't implant a healthy patient. And so sometimes what they'll do is they will have seizures during their time, and they'll just have like crazy spikes. And what someone that's processing the data will go in and do is they'll set it all to 0. And so here I'm using a little bit of domain knowledge to know that like this is not neural activity. Like if it's just like a flat line, that's not neural activity. But for all of us in this room, that's a pretty good guess. If it's a flat line, it's probably not neural activity. But that is also a good point where you all have domain expertise about whatever you're working with, whether or not it's flies or mice or rats or monkeys or humans. You should use that domain expertise to build rules for your data processing. Once you visualized it at a high level, then at a low level, and then once you have those rules, you don't have to look at your entire terabyte or 2 terabytes, or I think like Chen had like 40 terabytes worth of, or gigabytes maybe, never mind, 40 gigabytes worth of data that she was bringing today. Okay, does this make sense to everyone? Yeah.
Will be too early at this point to do some low pass filtering to give more texture to the soup?
Yeah, so this data set, what I'm visualizing here, it already was filtered.
Okay.
Yeah.
Forget about it.
Yeah. Okay, does this make sense?
Do you want to keep the rest of the neurons that are active, but it wasn't during epileptic seizures? So
Yeah, so because these electrodes are very far apart, I can go back. Because they're far apart, so like if I have, let's say I'm looking at the, let's just look at the green. If I have something that, depending on like the seizure, if I have something up here that's like, pretend there's like a focal point here, maybe these electrodes are corrupted. These ones down here, depending on the magnitude of the seizure, might not be. So that's a choice based on like the underlying biology of your animal. Yeah. Okay. All right. In the interest of time, we're going to have to breeze through these slides a little bit. Okay, the next one is signal extraction. And so here, I want to ask, what types of signals might you have in your data that you do not want in your data?
Noise.
Okay, good. Noise. That's kind of vague.
Blinking.
Okay, blinking. I heard something Nina said.
Oh, no, I made that joke.
Yeah.
Okay. EMG, okay. EMG, ECG, all right. Geeling, noise in data. So far we have blinking, EMG, ECG. Line noise, yes. Electrical noise, that's important, yeah.
You said that these were collected 24 hours, 7 days.
Yeah, so okay, so yes, this question is not necessarily targeted towards this data set. This is just generally in neuroscience. But yes, there will be examples on what I show you next that apply to this data set, yeah. Okay. All right, I think we all have the idea, but those of you that did not answer, I will call on you next, and there are many more questions. Okay, so some of these are lung artifacts. For instance, if I am breathing, my body is moving, and the electrodes in my body, depending on whatever you're recording with, or even images, like those of you that do calcium imaging, the microscope moves, like the brain moves, like these things are live. So breathing, heart artifacts like ECG, EKG, like you're gonna get a heartbeat in this data. Stimulation artifacts, like if you're working with human patients, or a lot of times like monkeys, what you're gonna do is like you might wanna stimulate because they are diseased and you're trying to get rid of it. Sometimes you can record when you are, and you're gonna get some stimulation artifacts. Another is movement artifacts. Like what happens if we did neuropixels recordings in like Marcus's lab. The animal is not comfortable. Like they are head fixed onto a thing. Like they're trying to move around. They want to get away. Like you have to be able to account for that, and you have to deal with it. Another one which Geeling noted was electrical noise, also known as line noise. And there are many more. This is just an example, and I'm sure you guys can come up with a list that is 10 times this long for all the types of noise you might encounter. Okay, so when we look at noise, sometimes the easiest way to view it is not in the time domain, but in the frequency domain. And so one of the most useful visualizations that I'm sure you guys are all familiar with are spectrograms. And so what I'm gonna do, and we're gonna breeze through these a little bit because we're only on our second principle, but this is when you look at different types of lung artifacts, and these are specifically for human and for diseases. You might not find this in your monkeys or your mice or your rats. But if you have different types of diseases, you're going to get different types of time series waves, and then you're going to get different types of spectrograms out of that. And the spectrogram is going to be on top of what your normal data would be. And so you're going to need to figure out a way to remove that data, either in let's say it's easier in spectrogram. Let's say there's just a band at 40 hertz. You're going to have to find a way to isolate it, remove it from your data, and then transfer it back to time, or maybe it's easier to remove in the time domain. Another option or another example is heart artifacts. So you can have many different types of problems with your heart. You can have many different types of time series, and that leads to many different types of spectrograms that you're going to get out of it. And so here what I want to really demonstrate is that there's a lot of things that happen with moving animals, a lot of problems, and you might not be able to identify them in the time domain. You may have an easier time identifying them in the spectrogram and in a spectrogram in the frequency domain. Okay, so here what we're going to do, this is a time series signal. And what I'm going to do is I'm going to add some noise to it. And so this might be a heartbeat. I think it's literally, I just plotted a sinusoid, but close enough to a heartbeat. And if I add them together, I'm going to get something like this. And does anyone want to make a statement about if I take the signal on the left and the signal on the right and I add them together? And this is what we get. What do people think? What is concerning about this to you? Yeah, you didn't see anything, right? So if I was just looking at the waterfall plots that I showed to you before, you wouldn't see anything. They look pretty much the same, right? If I was just looking at time series, I would look at this and be like, you know what, that's fine, that's good data. But if I look at the spectrogram, then this is the spectrogram only for that middle image, so just the clean time series, that's what I'm gonna see. Versus if I look at the spectrogram for the bottom image, that's what I see. And what you see is this line that comes up at around 20 hertz right here. And the reason is because you have this signal writing underneath, and it could be your heartbeat, it could be, line noise, it could be anything. And so definitely make sure, especially when you're thinking about signal extraction, to look not only in the time domain, but also in the frequency domain. Because you're gonna see things, and it may actually be easier to do rejection, like artifact rejection, in one domain versus another.
So
Yes?
we also know here that this band there is left there because we put it there. If you see a band like that, just the fact that it's so perfectly continuous,
Yeah.
that's enough for you to know that it's an artifact?
Yeah, so I mean, this kind of goes back to Amit's question before about like, how do you know that this is bad? And part of it is gonna be the domain knowledge that you guys have. Like, you all are experimentalists. You know exactly what is good and what is bad, whether or not it be intuition or like principles that you've actually written down. And I would recommend you all write down your intuition as principles. But when I'm looking at this, and I know that I have something like a heartbeat, which is going to show up around, this frequency band, I'm going to be questioning it.
But I guess more broadly,
Yeah.
like if you're doing something completely novel, right?
Yeah.
Just the fact that all the other frequencies, they have a time component.
Yeah.
And this one just sticks out by having no
Yeah.
time. That's perhaps something that can inform you as a biologist, going biological data, that's
Yeah, so I mean, I definitely agree. The idea that like, no, I do agree that if I see something, I'm gonna go back actually to the time series one just to look at it. If I see something, okay, maybe one thing that you can do if you don't know is you have to, and let's say there's no ground truth, you've never done this experiment before, compare to what the other biological signals look like, to what your other animals look like, to what other electrodes look like, to what other like images from a mini scope doing calcium imaging look like. Because if we're looking at the waterfall plot, if I'm looking at all those time series and then I see just this like thing that sticks out with like zeros or like a line, I don't think that that's right. Even if I don't know anything about that data, that doesn't look right to me. And if I go back and let's say I had 100 instances of time series that looked like they were on the top and time series that looked like on the bottom, and I'm just looking at it, the thing on the top, let's say I have 20 of them and something like this at the bottom, it just doesn't look right. Yeah. And so that may not be satisfying because it's not something, it's gonna be very domain specific and it changes for like the animal model or like even their conditions. But you know what you're gonna expect. You're not gonna expect lines. Yeah, okay. All right, let's keep going. Okay, the next thing I wanna look at is data set augmentation, and we're gonna actually shift out of time series domain and we're gonna talk about some images for those of you that do imaging data. But first, what is data augmentation? Ideas? It's okay if it's half-baked. But actually, I'm going to call on someone. Okay. Laura, what is data augmentation?
When you increase the amount of data that you have from your existing data.
Okay. Okay. So did everybody hear that? When you increase the amount of data that you have by adding data. Okay, so that's actually a pretty good stab at it. So data augmentation and data analysis are techniques used to increase the amount of data by adding slightly modified copies of already existing data or newly created synthetic data from existing data. And so I actually pulled this from Wikipedia, and I actually really like this definition. I think it's pretty good. So there are ways to do data set augmentation in every domain. It may not be the easiest. It may be easier in images than it is in time, or easier even still in like single cell seq. But there are ways to add data to your data set. Let's say you don't have enough because one label or one class, it's just too little and you can't learn with it. So you should think about how can I tweak my data to actually add to it. Another thing I wanted to point out about this definition, or perhaps add to the definition, is that it acts as a regularizer and helps reduce overfitting when training a machine learning model. Now, who wants to tell me what I mean by this statement? Yeah.
I'm not totally sure what you mean, but there is, well, I don't think I've done data augmentation, but I think what I'm understanding, the idea is that you're taking the properties by producing more of that, in a sense, you are creating a data set that, well, is also noisier, right? So it would prevent overfitting.
Yeah. No, that's completely right. Basically, by taking the data, making it a little bit different, tweaking it some, you're forcing a model to learn more about what the data is versus what noise is. So you're making it invariant, and that's an important word that you guys need to know. You're making it invariant to whatever type of augmentation you're adding. Okay. All right. Now this is an image of 3 cells. One of them, and they're dyed to have different things. So one of them is like, let's say, dyed to have the nucleus be very apparent. One is dyed to have the cytoplasm be apparent, and one is dyed to have the mitochondria be apparent. If I give you this image, let's say we're doing, fluorescence microscopy. How many cells are in this image? How many cells are in this image? The image on the right. All right, does everyone think it's 13?
Seems like
Unless every single one is
It seems
supposed
like
to
a
be.
trick question. Can we count?
I'm sure you can count.
What is this image?
What is this
An
image?
image from a slide?
Yeah, it's an image from a slide. Well, I mean, this is not an image from a slide.
But it represents
I have
it.
put up an image of cells. That's all I'm saying. I've put up an image of cells.
No, but it's a cartoon. first of all.
It's a cartoon. Oh, fine.
It's a
I've
drawing.
put up a cartoon
Yeah.
of cells. How many cells are in this image?
Unique cells?
How many cells are in this image?
Well, by definition, you didn't say unique, so we just count it.
Okay. All right, fine. I think you guys are getting the point. There are 13, if that's what you counted. I didn't actually count. There are 13 perhaps cells in this image, but there are 3 unique cells in this image. I've taken the 3 cells on the left and I've put them there and I've just changed the orientation. I've rotated them. Of course, it's a trick question. I'm trying to get you guys to think and learn. Basically, the idea here is that the blue cell where I've like highlighted the nucleus, it doesn't change whether I rotate it. It's the same cell. It doesn't make a difference. Same with whether or not I've highlighted the cytoplasm or the mitochondria. The cells have not changed. And if I had an image processing pipeline and the goal was to recognize cells that had nuclear staining, cytoplasmic staining, or mitochondrial staining, I wouldn't want my computer vision pipeline to only recognize cells in like the orientation that you see on the left. I want it to recognize cells in like all of the orientations that you could ever possibly see the cell. And so this technique in data augmentation is literally just rotation. You take an image and you rotate it. And so this would be an example of, however, oftentimes the most common thing is you just rotate by 90 degrees and you get 4 different examples of the exact same image. But this is an instance in how you can increase the amount of your data by a drastic amount such that your model will be invariant to the rotation of the image. And so just the concept of data augmentation and taking the same thing, just rotating it a little bit to make your model invariant, does that make sense to everyone? Yes? Okay, good. All right, the next thing that we're gonna do is normalization and standardization, and this is going to be very domain specific. The concept applies across all, but obviously the way you do it, and perhaps sometimes even if you should do it, is very dependent upon the data. Okay, so first off, what is data set normalization? UA.
It depends on what's the goal of normalization. For example, if in the data you have different variables that are using different units, then you want them to be comparable in a high-dimensional space, then you normalize them by maybe z-scoring to have the same mean over all and same standard deviation.
Yeah, no, that's good. So z-scoring is good. And I think you've answered the question. You've answered a question 2 and 3 layers deep of why should I do normalization or what is normalization? So I think that that's good. She was more thorough than I wanted, but that's fine. So normalization, just at a higher level than what UA gave us, is adjusting values measured on different scales to a notationally common scale. So the idea is, let's say, and I already showed you guys this, I've got 12 patients. They might have very different activation levels, but the idea is not so much the true activation level, it's the relative between conditions, let's say, between these move rest conditions. So I want to put them on the same scale such that when I model them, my model isn't picking up this notion of these true scale. It's picking up the relative differences between a move and a rest condition. Okay, so the first part is to put them on notationally common scales. A more sophisticated thing you can do, which we are not going to go to today, is to take probability distributions of adjusted values and put them into alignment. So this is the idea of normalization. Okay, standardization, which oftentimes is thought of as a special case of normalization, it's when you subtract the population mean from an individual raw score and then divide by the population standard deviation. And UA actually already mentioned this one, which she gave a much more thorough answer than I wanted. This is also known as z-scoring. And I wanted to highlight this because in neuroscience, a lot of people use z-scoring. And so you should all be very aware of what z-scoring is. And just to give some, oh no, I had an equation, but I guess it didn't show up. I will update the slides. Does it show up on your guys'? No, maybe? Okay. Anyways, I'll put it such that the equation shows up. You take an instance, so the equation is z, which is the z-scored value, equals x minus the mean over the standard deviation, where the mean is the population level mean and the standard deviation is the population level standard deviation. So z equals x minus mean over standard deviation. And I will update the slides to have that. Yes?
In this case, you will be doing that per electrode, per patient. Right?
Yes. So in this case, what I would do is you've got patient days electrodes across all time series. Mm-hmm. So what I would do is I would do it on a per patient, per day, per electrode base. Yeah. Because, so, and we're actually going to get to this. So good segue. What does this help us correct for in our data? What does normalizing or standardizing help us correct for?
Just being on the different baseline.
Yeah. Yeah, so different baselines for sure, but there are different ways to be on, there are different ways to have different baselines, right?
Yeah, it
So
will be your instrument.
Yeah.
They will be using different instruments.
Yeah, that's a good one. Okay, Manchuan, how about you? What are different, why would we do this? So Gregorios' answer was people have different instruments across, different labs, and you want to normalize, standardize for that. What else might you do it for?
So different people, probably they would like the signal, levels would be different.
Yeah. So that's a good one. Patience is another good one. Changying, how about you?
I think sometimes maybe the signals have different distributions, like sometimes a little bit have a bigger variance or something.
Mm-hmm.
Want to make them of the same scale.
Yeah. No, that's good. So that's kind of getting at the idea of habituation. Let's say you're doing an experiment for 3 hours and the mouse has learned it by the end. There's still a difference between the conditions, but there's habituation. So these are all important ideas that we need to think about. Yes, Nina?
I was going to say, also for statistical analysis,
Yeah.
you're comparing several variables and you want them to be on the same scale.
Yeah, absolutely. So all of these are good points. These are just a few, and you guys covered most of them, if not all of them, but neural population drift across time. And this can mean physical, electrode shift across days. So both of them, so neural population drift, that can lead to the underlying representation of what the population is responsible for. It changes across time. We all know there's plasticity. There's, if you have a maze, you've got different mazes that you're learning. One electrode, it one day might represent left turns in a maze. I know I'm drastically simplifying this. JU's probably pissed, but that's fine. And then a month later, it is responsible for right turns. So very simplification, but that's the idea. Electrodes shift across days. Now this shift is not the population representing something different. This shift is physically electrodes move. And so when you think about normalization or standardization, think about it in the different dimensions that can occur. It doesn't just occur across time. It doesn't just occur across days or months or seconds. It occurs physically too, like spatially as well. Another, which Chen got to, which was Man Chuang got to this one, different dynamic ranges across patients. Like my brain is very different than Brady's brain, which is very different than Matt's brain. It's all very different. But you're gonna have to account for that, especially if you want to model all patients together, and we'll get to this a little bit in the model engineering lecture. And another one, this was the one Chen got to, which was habituation to stimuli. Like if you're doing the same experiment for like 5 hours, the mouse is gonna have a very different reaction in the beginning than it does at the end. That doesn't mean there's a difference between the we had mice with neuropixels. We showed them like videos of predators versus prey. Like there was a stark difference at the beginning. 3 hours later, there's still a difference, but it's just not as big in magnitude. So you need to account for all of this. Okay, we've got 6 minutes. Oh, and I have an example. Okay, so we'll do this quickly. So this is the data that I showed you before, zooming in to the 25 the 25 instances across time for one electrode. And here what we've done is we've gone ahead and we've actually standardized our data. So it's still a little bit soupy, as I think was Gregorios's language, but you can see a little bit clearer that there's just something up that's questionable. And if we look at our waterfall plots, you can see that the ranges are a little bit all over the place. And of course, we're gonna get to like, oh, how did I actually plot this? What were the scales I actually used? And if you want to do that, you will work on this data today. But if I then go plot it, the standardized version, you can see something that looks like this.
This is Z-scoring?
This is Z-scoring, yes. And so for a lot of the trends that you see, like let's just look at this bottom one, you've got this dip and then it comes up and then you've got a dip again and then et cetera, and you kind of see the same thing here. Okay, yes, Jay.
Standardized to the mean of each one of these traces and the variance of each one of the traces for the population.
Yes, so this is a good point, and I was actually going to ask a question about this. This is to each of the electrodes. You can't do it to all of the electrodes because the electrodes are in very different locations. So what I've done is I've taken some piece of the time series for each of the electrodes, and I've found the population mean for that piece and the standard deviation for that piece. How much time did I use?
I mean,
This is not a theoretical question. You can actually tell me how much time I used.
well, I don't know, I can guess what you did, but I would guess that you want to, if you're going to do it by day, you want to do the whole day.
No, so this isn't by day. I'm just taking the example we had before where it's like I fixed a patient, I pulled a day, I pulled an electrode, I have all my time series instances, and I'm just gonna standardize or normalize.
I would take time to know around the behavior.
Sure, you can do that, although we did already kind of discuss how this is naturalistic data. And so,
Like,
yeah, so this iteration of it is the naturalistic.
So you can take the whole day?
So there's also drift across day though, right?
So,
But okay, so you guys are getting into like a higher level discussion. You can literally, by looking at this slide, tell me how much time I used to do this population standardization.
1,000 of whatever.
Okay, why?
800.
Why 1,000? Why 800? You guys got
Sorry,
different.
Is it 900, like in the bottom graph? No. Forget about it.
Okay, you guys are getting at the right idea. Okay, so what I'm gonna demonstrate here is that this is the original data. Is actually 1,001 time steps, but let's just call it 1,000. And that's 1,000. Here, when I plot it, this is 900. Why is it different?
You took, what did you take?
All right, we literally have 3 minutes and we have not covered the last topic yet, so I'm just gonna tell you, although this would be more Socratic if we had time. What I did here is I took the first 100 time steps right here. because it was like just the beginning, there is no behavior here, it's just naturalistic, it's like a rolling thing. I took the first 100 time steps for each, so I think I actually did this wrong, but that's okay. It's fine. I won't tell you how I did it wrong, you'll tell me how I did it wrong. What I actually did is I took the first 100 time steps and I found the mean and the standard deviation for the first 100 time steps. And then what I did is for the rest of this time series instance, actually no, I didn't do it wrong. For the rest of this time series instance, I subtracted off the mean and the standard deviation that I calculated in this section. And then what I'm plotting here is literally that standardized like value.
Why wouldn't you take the whole thing, or if there is data you say, when you have like a moving window where...
Yeah, so you can have a moving window. And so I actually did do this right. I said I did it wrong, I did do it right. You can have a moving window, but it depends how you want to process your data. If you want to process your data in like chunks, I think this is like a minute and a half, given the sampling rate, you guys will get into it. So you can do it that way, but then what you're gonna have is like overlapping time segments. So if I had a moving window, like I would not have just this.
Why did you just take, you didn't take the whole thing because you're worried about the drift?
So I didn't, what do you mean the whole thing?
Like why do you take just the whole...
Find the mean and the standard deviation for the whole thing?
Yeah.
And then subtract the mean and the standard deviation for the whole thing across that one instance?
No, I guess, yeah, for every time point.
Yeah.
Like right now, you're going and the mean you're subtracting is the mean of the first 100.
The first 100, yeah.
Why are you not subtracting the mean of all of it?
So the reason that I'm not subtracting the mean of all of it is because what you want to do is you actually, and this is an important point, which we will get into if you do this data, you don't necessarily want to, and this kind of goes back to the modeling, and so perhaps I should have put this in the modeling section. You don't necessarily want to contaminate your trained test data with whatever you're using for normalization and standardization. And so here, it becomes a little complicated. So I might have these number of instances. I might have like, this is just 25 instances across like 1,000 time steps. I need to have this in a train set and I need to have this in a test set when I run a model. I can't use like the mean, or it depends how you want to do it. You can't always use the mean and the standard deviation for your test set to correct your test set data. Because the whole purpose of your processing pipeline is that you're not like implicitly baking information about the data that you're using to test into your testing data.
Well, in that case, you exclude, let's say, 10% of your data for the test, but you should still be using all of the rest.
Right. So here what I would do for this like instance, and what I would have done is, let's say I used like another 25 instances for test. I wouldn't take the mean and standard deviation of those. I would use these means and standard deviation.
All of the...
You can do it. I mean, we can talk about, so the reason that it's this way is because it's like a naturalistic data set. And so because there's not like clear move and rest examples.
You don't think it's that important?
Well, it's also because there's like drift across days. And so we want, because like,
That's also gonna help you with that, right?
well, it will, because you have like move and then you might have watching TV and then you might have eating. And so because you have different like behaviors that are like one after the other, you don't actually want to normalize and standardize with the eating for what you'd get with like TV. You want it to be a little bit of the beginning of each of those behaviors. Okay, but we're already, okay, does that make sense? We'll talk more about it. We'll talk more about it. Okay, I'm a little bit over, but we'll just keep going. We'll go very quickly through this next one. Okay, so you guys have done this already. Actually, okay, you've talked about this concept already, and if you want to dive into this more, there is a notebook that you have not yet finished. Okay, so this is the idea of train validation test splits, and this will bleed into the next lecture on model engineering. But basically the idea is that you remember on the first day, what we ended up doing was the single mouse data set where you had the time series and you had a time series, and then across the time series you had different visitors in the cage. You had a female mouse and a male mouse. But who can tell me what, like, so the female mouse and the male mouse, they did not have the same like number of time series instances. The classes weren't balanced. But what was the other behavior that you guys had that was like even more severely unbalanced? Attack. Perfect. Okay, let me do this. All right, we're not going to go in presentation view, but that's fine. I'm going to scroll. Okay, here we go. Okay. So yes, so this was the data set, just for those of you who don't remember it. We had a mouse, we had a visitor in a cage. We went ahead, we took this data, we visualized it into 3 principal components. We plotted it against time, and we also plotted it against attack. So one of the things that we noticed, and I think you may have done this in the second notebook, but one of the things you noticed is that attack versus the other like behavior label, it was not balanced. They were very unbalanced. And so when you actually did your train set and your test set with regression, you never predicted anything for attack because you didn't really have that many examples of attack. And so this is a third notebook that we didn't get to on that day, but if you want to dive into this, that notebook is there and you are welcome to do that as well. Another thing that you can do is that here, what we did and what we wanted to try, which we didn't get to, you can actually just take out other labels. Just make the classes balanced. And you'll have the same number of attack labels and the same number of other data labels. And so here what we're doing is we're just plotting the fact that we have the same, we're plotting them in principal components just so that you can see it. But once you actually balance your number of labels across your train set, and in this case your test set, we don't have a validation set, what you see is that you actually start to get a better job of classifying your train and your test. And so making the classes balanced by deleting data is one way to go about this. Another way to go about this is to change your loss functions such that you're penalized for making a guess of the larger class, so that the model knows that, oh, I do get penalized for just guessing this. So there are many ways to address this problem. This is just one of the ways that we addressed it, and you can dive into the notebook more if you want this. So this is actually the notebook that we did not get a chance to get to. So you're welcome to do that as well. But are there any questions about this train test validation split? And making sure your classes are balanced across these different splits? No? Okay, good. With that, let's actually go back to the top. I'm just going to run through these 5 one more time so that you guys, we say them. So today, you're all going to be diving into doing it with your own data. Make sure you visualize it, develop an intuition with it, do signal extraction or denoising. Maybe it's already done for you. Augment your data if it's valuable and if you can, and if it's important. Sometimes you don't need to. Then normalize, standardize your data. And lastly, you can start to make some train validation test splits such that when we get to the modeling section, you're kind of ready to go. Okay? All right, let's do it.